And I'm excited to announce that next up we have Hendy from the University of York, who will be talking to us about the applications of paleoproteomics. Take it away, Jessie.
Hi, thank you. Sorry, just fiddling with Zoom a little bit. Yeah, thank you so much. It's such a pleasure to come and speak today. And I'm sorry, I've got a little bit of a cold, so I've got this weird husky voice going on. But yeah, it's a real honor to be asked to talk about the applications of paleoproteomics. Quite a big ask, I have to say, but I'm really excited about it. And I'm just thrilled to hear from all the talks for the rest of the day. And thank you so much to the organizers for putting this together. So yeah, I'm Jessica Hendy. I'm based at the University of York in the Bioarc Research Facility, and I'm going to talk about applications of paleoproteomics. And just to note that this presentation does contain just a few images of human remains. So we are so lucky in this field to be able to work on such a huge diversity of applications. There is such a myriad of applications for shotgun proteomics. So by paleoproteomics, I'll just quickly clarify, I'm basically talking about the use of shotgun proteomics or LC-MS/MS for ancient protein studies. Maybe a little bit inaccurately, but for the purposes of this presentation, that's how I'm defining it. So as you can see, there's such a huge diversity of applications, and I'm not going to read these all out, but in my presentation today, I'm going to focus on just a few of them. And this diversity is also reflected in the kinds of materials that we can analyze. So human and animal remains, whether that be, say, bone, but also mummified remains, food residues, a range of different kinds of organic artifacts. So I hope I can sort of convince you of this diversity. The way that I'm going to do that is take you on a sort of global grand tour and just highlight 5 examples, which I hope encapsulates this diversity in method, but diversity in sample type. So first of all, I'm going to take you to the high altitudes of Tibet and talk about how paleoproteomics is used in studies of human evolution. So this is high altitude Tibet, and within this valley system, cave yielded a really fascinating mandible. So it's a cave that's still used in the Buddhist faith today. And this mandible was dated by uranium thorium dating to around 160,000 years ago. Ancient DNA was tested, but there was no ancient DNA survival. Similarly, no proteins found in bone, but proteins found in dentine, and these were highly degraded, as would be typical for ancient proteins. And phylogenetic analysis of these protein sequences showed that this individual was most closely related to Denisovans. So Denisovans at this point had only been discovered in Denisova Cave or in the sort of genetic legacy that they left behind in subsequent populations. So a hugely groundbreaking study. So what this demonstrated is that archaic hominins indeed occupied the Tibetan plateau and indeed were adapted to these high altitude environments, though of course this individual did die there, so but they in general were seem to be adapted. This is 120,000 years before the next sort of oldest known Paleolithic site, so hugely groundbreaking for that archaeology. So I think what this showcases in terms of applications of paleoproteomics is that one avenue is that it can enable phylogenetic studies, and it can be applied beyond the limits of DNA preservation, which makes it really exciting for different kinds of contexts and also in deeper time periods. So now I want to take you to Jordan and talk about how paleoproteomics can be applied to the understanding of paleo environments. So what I mean by this is how paleoproteomics can be used to understand paleo-environmental reconstruction, but also a broad blanket term maybe is animal and human interactions. And Sam, in her talk on Zooms, obviously touched on this as well. And the overall principle behind this really is that animals can occupy particular ecological niches. And therefore, the identification of particular taxa can be achieved by analyzing protein sequences, as Sam has mentioned with Zooms. And this could be fauna remains, this could be material culture. So for example, if we find this lovely Viking comb in, say, rural Yorkshire, and we discover it's from reindeer, that would suggest that there are long distance trade routes or movement of these objects in the Viking world, which we do know indeed to be true. So a lot of these applications come from zooms, which is brilliant, but sometimes paleoproteomics can help here by perhaps verifying zooms markers, or if there's a scenario where perhaps zooms isn't quite able to get that species level distinction, but that might be really important for your research question. A full analysis of protein sequences of, say, collagen or other proteins might enable that species identification. So I didn't want to talk about chromes, I want to talk about birds. So thanks, Claire. And talk about the site of Shubeika in Jordan. So many of you might know this site. It's a late Paleolithic, Neolithic transition site in Jordan. And many of you might know it because it yielded evidence for what seems to be very early bread-like products, which were detected via SEM analysis of some preserved bread-like remains. So a really exciting finding in its own right. But I want to highlight the study of lysiomens, which analyzed the eggshells found at the site. So the site stretches from the Natuvian all the way to the pre-Proterozoic Neolithic, and it spans the Younger Dryas, so a time period which people think is cooler and drier in general in this part of the world. So in the study, they analyzed 35 eggshell fragments from 2 periods of the site using peptide mass fingerprinting and also paleoproteomics on a few specimens, and found that there were ducks, geese, and swans all breeding near Shubeika, because the eggshells were found there. So what this means is that water must have been present at least at the time that the time of egg hatching, so at least between spring and summer, and even during the period of the Younger Dryas. So really critical interpretations for not just how people were using the site and the interaction with these animals, but also insights into the local climate. So in terms of how paleoproteomics is being showcased here, it's a really amazing example of how paleoproteomics is coupled to modern ecological data and the value of integrating zooms and paleoproteomics together. So I now want to take you to Peru and discuss applications in sex and gender. So many of you might know this study, which was published a few years ago, which analyzed 2 individuals that were found in southern Peru. And essentially what this study was based on is amelogenin peptides. So amelogenin is a protein and its gene is found on the X and Y chromosome, and therefore it has X isoforms and Y isoforms. And sequence differences between these 2 isoforms mean that we can detect the Y form, i.e. the presence of a Y chromosome, or the X form, the presence of an X chromosome. So in this study from southern Peru, 2 individuals were analyzed for amelogenin in their enamel. These were individuals that were associated with paraphernalia from hunting big game. So essentially, stone tools from big game hunting. And one of these individuals was male and one of them was female. So this is in contrast to a hypothesis that was originally put forward in the 60s that it's primarily men that are being hunters and hunter-gatherer societies. There is other research that suggests that this is not necessarily true, but in this case study, at least, this is really questioning this long-standing belief. But like I said, there are ongoing archaeological arguments that show that man the hunter is indeed perhaps an outdated concept. So what I'm particularly excited about in the field of amelogenin studies is the advances in mass spectrometry that's going on in this area. And so I just wanted to highlight 3 papers that just all come from 2024, which is amazing, that are all about trying to reduce mass spectrometry time and therefore cost for amelogenin analysis. So sorry, this is a bit of a wall of text here, but you can see a lot of efforts both in the lab and in the mass spec to really reduce the cost of this analysis. And this has real ramifications, because by decreasing the cost, you increase scale, and this leads to completely new questions that you can ask with this method. So now, we might have the opportunity to look at things like mass graves, whether there was, say, sex selectivity with particular diseases, things like patterns of infant mortality with a degree of statistical confidence that we might not have had before. So just to highlight this paper from 2022, which looked at a rather large assemblage of children, non-adults from Bronze Age Austria. And what this showed is that there was gendering of these male and female children that was the same gendering patterns as the adults, as shown by grave orientations and grave goods. And what this shows in this plot, by looking at sex estimation in line with age at death, it shows that there is some vulnerability of these young male infants, it appears like. So these younger male infants seem to be dying more so than the female infants at this point, which is a really interesting observation. But I think it is worthwhile bearing in mind a couple of points to do with the melagenetic analysis, that so far this method is only looking at these binary karyotypes, XX and XY, and that chromosomes alone are not necessarily the only determinants of what some people call biological sex. And then, you know, it's a long-standing archaeological question that is there an accurate link between expressions of gender in graves and sex? So let's just not ignore those arguments, I think. But why this method is so cool, I think, is that it's cheaper, it's faster, it's less destructive than ancient DNA methods, if indeed your question is about sex estimation. And it is so ripe for integration with other methods. So for example, with paleopathology, looking at patterns of disease in the past and how those might be sex specific. There's so much potential there. So now I want to take you on a fourth case study to Alaska and talk about how paleoproteomics can be applied to studies of disease and physiology. So this is Nunalak. It's a pre-contact site in coastal Alaska. And sorry, I just want to highlight the amazing project website from nunalak.org. It's the best archaeological project website I've ever seen. It's really interactive, and I really encourage you to go look at it. It's really beautiful. So at the site is amazing organic preservation. For example, many fascinating wooden objects, but also at the site were paleofeces recovered. And a PhD student that I helped supervise, AK, was able to analyze 5 of these paleofeces, which she published in 2021. So the goal here of the study was really to identify the host origin, for want of a better term, of the feces, to identify any consumed foodstuffs within the animal from the feces. And then this study applied a particular methodology that was deliberately targeting eukaryotes. It wasn't sort of targeting gastrointestinal microbiota, which is a sort of deliberate choice in the extraction method following a previous publication. So what this analysis revealed is peptides that could be assigned to dog. So therefore, we think that the dog is the origin of the depositor, again, for want of a better word. And what was interesting is that there was also peptides associated with salmon. And this is in line with archaeological evidence at the site. Zoom's analysis was also applied because there were tiny bone fragments within the feces. These were also salmon. So we're able to identify the host origin, but also what these animals consumed. What was really interesting is that with the proteins that were from the salmon, we could see that these were proteins that were found in the gastrointestinal tract of the salmon. So we suspect what might be happening is that the fish were being gutted, and then their entrails were being consumed by dogs. So this is maybe giving us some insight into actually how people themselves were processing the fish. So just to look at this data in a slightly different way, you can see that there is really a range of different, I guess, preservation or protein recovery, I should say, between these different specimens. So you can see that in one sample, there wasn't really anything endogenous that we could see there. So this is just highlighting that I think there's still a lot of research to do when it comes to paleofeces analysis with protein research, but it's really exciting. So what did we see with the dog proteins? We saw indeed proteins and a snapshot of a proteome that was associated with the gastrointestinal system. So we could see proteins that were, say, expressed in the small intestine or the colon. And we could see proteins that were playing roles in fat and protein digestion and also the mucus layers of the GI tract. So in future, I think this could be really exciting for looking at things like gastrointestinal physiology and disease. But like I said, there's still a lot of groundwork to do here. So I think in terms of showcasing paleoproteomics, what this study does is that it really shows these multi-species insights. So we're getting insight not just to the dog, but also to the salmon, but also how humans were interacting with these animals. And this study also exploits the fact that proteins can be expressed in particular tissues, and they also play specific roles, and therefore that gives us insight into our archaeological sample. So, and now I want to take you home to York, and I want to talk about beer. So, as Bayer mentioned, I work quite a lot on food proteins in archaeology, and obviously protein is a major component of food, so it sort of naturally lends itself to that analysis. And there's a whole range of contexts in which food proteins can be recovered. So, for example, in the sort of top line here, we've got very well preserved context, either very arid context or preserved in sort of bog-like conditions, waterlogged conditions. But also in things like dental calculus, we see some evidence of food, and also on food residues. But a substrate that archaeologists have been reporting for quite some time is something called beer stone, and this is probably calcium oxalate. And so this is Lindsay Paskillen, and she, as part of her master's research here at York, was able to analyze a sample of modern beer stone to just see what kinds of proteins do we see in this. So the very kind people from York Brewery gave us a sample of their beer stone from the fermentation tank of Guzzler, this very delicious pale ale. And I promise you, they do clean their tanks, which is why we were given an access to this sample. This is a very commonly occurring phenomenon to have this beer stone mineral accumulate in fermentation tanks. So what she found is that there was indeed that beer stone does entrap proteins that are reflective of this product. So she found an abundance of evidence of yeast, and also of barley grain proteins, and also some wheat, which we know was part of the recipe of this particular pale ale. But interestingly, she also found no evidence of hops, even though we knew that was in the recipe. And so, yes, while indeed this beer stone mineral does entrap the signature to some extent, doesn't necessarily encapsulate its full ingredients. And this diagram really highlights, at least in a modern beer making process, all the myriad of steps that go on to create the final product. And if you think about what might then be deposited in an archaeological record, you might only have a few of these, if any of them. So you really need to think critically, I think, for any paleoproteomic study, but particularly for food, about this palimpsest, the chaîne au potoir, if you like, of processes that are going on that you might not even be aware of. So this is something I really want to flag, especially when it comes to thinking about the value of experimental archaeology for tackling some of these questions. So, for example, recently, Miranda Evans published a study looking at experimental food crusts and ceramics, cooking different ingredients, and then looking at their recovery. So what she found is that not all ingredients were detected equally. And, following on what Bayer said, this isn't wholly surprising in terms of what we know about protein diagenesis, but really important to bear in mind the discrepancy between those sort of input ingredients and then our final detection. So to summarize about this application of paleoproteomics, there's this incredibly rich potential for food proteins from quite a wide range of archaeological sources. But I would really stress the value of something like experimental archaeology for identifying these biases, which are really critical for making accurate interpretations. So I want to thank you for being on this global tour with me, and I'm happy to answer any questions that you might have, either now or I've put my email address down there at the bottom if you prefer to get in touch by email. Thank you so much.